Thank you very much. We have really heard really fantastic presentations this morning and many different perspectives. And maybe one of the things that I was thinking about is that when I finish talking, that maybe all of us that talk this morning, we can field the questions from the audience, and maybe we could elicit some discussion about what we talked about. So I just want to provide a different perspective, perhaps from the other side of the Atlantic. So we talked about the fact that there's a tremendous amount of interest in the United States and elsewhere because President Obama talked about precision medicine last year, and when leader of the country, talks about precision medicine in the State of the Union address, that's actually very significant. And although we were expecting that this is going to result in a lot of money for cancer research or other types of research, that didn't quite happen. But in any case, it really moral support for the kinds of efforts that we have heard, yesterday and this morning. And in this year, in 2016, President Obama talked about, a special effort in the United States about cancer. And the goal of the effort that he outlined is to try to think about, cancer and the developments in cancer and how we might be able to accelerate, the progress that we could make in cancer. And the goal is to achieve goals that usually we achieved in 10 years within a 5-year period of time. And he asked Vice President Biden to actually lead this effort. And so the goals, for cancer is to be able to, obviously, many of the things that we talked about. How can we accelerate the design and testing of effective treatments tailored for individual patients? And how do we expand genetically based clinical cancer trials? This is a very important issue that all the speakers this morning, talked about. They also talked about establishing cancer knowledge network. Just, although each of us would think about our own continents and so on and so forth, we should probably be thinking about this worldwide as to how we might be able to bring together all of these types of information. And with all of these goals in mind, Vice President Biden put together a blue ribbon panel to think about, and make recommendations about what are the kinds of things to do. And that is called the U.S. Moonshot Program. And the report from this Moonshot Program just came in about a couple of weeks ago. And these are the recommendations that are made by the blue ribbon panel. And as David Cameron talked about this morning, one of the first and important things is to be able to try to establish a network for direct patient involvement. The patients, usually considered as subjects, for clinical trials, and they hate the term of being subjects. And they want to be patients, and they want to be involved, as David talked about, and say, how do we think about getting, patients directly involved in them? The second is immunotherapy, and as all of you know, there's a tremendous amount of excitement about immunotherapy. Even though in any given types of cancers, the number of percentage of patients that are benefiting from immune therapy is 20 to 25%. There's still a lot of excitement about how we'd be able to manipulate, that outcome to be able to increase that. And again, as we heard this morning, we're to talk about, many of the targeted therapies, many of the patients, not all of them, develop resistance. And it would be great for us to understand the mechanisms of resistance to therapy, so we would be able to first be prepared that a patient might become resistant and be able to develop a strategy, or when actually a patient becomes resistant to a particular therapy, that we have alternatives. And there are a number of other sorts of things that also include, prevention and early detection strategies. And so there are a number of different types of things. There are already efforts currently underway in all of these areas, both in the United States and in Europe. And I wanted just to talk about a few examples of some of these types of things. And I'll begin with people keeping healthy. Especially in the case of cancer, as we all recognize, the cost of treating a cancer patient is very high. And number two is that if you diagnose a patient with a late stage of cancer, then the overall chance of survival for those patients is actually quite low. Whereas if you either prevent the development of cancer, or they actually be able to identify cancer at its early stages, the chances that we'd be able to effectively treat those patients is very high. So many people sort of think about having a good dietary habits and exercise and not eating red meat and cookies is going to help you, and that certainly does help. But there are clearly other types of factors that are very important. And so first thing that we need to think about is that within the population of people, how do we assess risk for cancer? And so one of those things that we talked about for the long example that we have known for a long period of time was the testing for breast cancer susceptibility. This has been around for a long period of time. And even though not all of the people who might be able to benefit from such testing have actually undergone such testing, but when somebody like Angelina Jolie talked about her own experience and her susceptibility for breast cancer, and the op-ed that she has written in New York Times, certainly in the North America has made a significant amount of impact. And as a result of that there was a significant increase in the number of patients who get tested for BRCA1 and 2. And so it's called the Angelina effect. But what is really important about this is that for many, many years, when you think about breast cancer, the only genes that we were thinking about for BRCA1 and BRCA2 testing. However, in the last several years, we realized that not only BRCA1 and 2, but a number of other genes which are involved in DNA double-strand break repair, mutations in all of those genes can cause breast cancer susceptibility. So when we think about testing for breast cancer susceptibility, it is not sufficient to be able to do only BRCA1 and 2 testing, but for all of these other sets of genes. Similarly, this is true not only for breast cancer, but there are a number of other types of genes in many, many different types of cancers that we could test now. Actually, as a result of that, recent studies have revealed that if you look at all of the so-called sporadic cancers that we look at, about 10% of them are the results of actually germline mutations in those patients. So that means that actually it is possible to be able to identify these 10% of the patients who are at risk for developing cancer by testing their DNA very early. Right? Another question that is associated with this, that many people say, okay, if you do all of this testing, what do you do with that information? Like in the case of breast cancer, there's a long debate about, if you identify an individual with a BRCA1/2 mutation, what do you do? And everybody says, okay, you increase the surveillance, for these patients. But many people ask, when do you initiate such surveillance? What is the cost of doing that? And how should we manage these individuals? Fortunately, there are studies now, to identify, the risk level for each of these mutations in each of these different genes, and we'd be able to develop guidelines as to when, in their lifetime you actually, start active surveillance. So one approach certainly is to be able to do this type of testing. It is not only for these adult onset cancers, but it turns out that recent studies have revealed that in pediatric cancer, about 10% of those pediatric cancers are the results of germline mutations. So if you look at the total number of genes that are known to be involved in cancer predisposition, there are now about 115 genes that have been described. So it is indeed possible to be able to do the testing for all of these cancer predisposition genes and Since germline testing is not that expensive and the cost of DNA sequencing are going down very significantly, I would like to argue that it would actually greatly benefit, being able to do this type of testing very early in the lives of the entire populations. And certainly that's a very controversial view that we could talk about the merits of such a view after I finish the talk. All right, so that is predisposition. Are the technologies that are available to be able to do early detection? And as I mentioned to you, in many types of cancers, if you detect cancer at stage 1 or 2, the long-term survival for those patients is nearly 90 to 95%. If you identify patients at stage 3 or stage 4, their long-term survival is only about 5%. So early detection would help. So but how do you do early detection? And one of the approaches that people have been thinking about is to try to identify whether there are protein biomarkers that might be present in the circulating plasma. So several years ago, we started a program to try to identify, see whether it is possible to identify biomarkers. And the approaches that we have taken to do that is dual. One is to develop mouse models, genetically engineered mouse models for different types of cancer, and then examine their plasma content before and after they develop the tumors, and assess whether there are any markers that can be identified in the early stages of tumor. And the second is actually try to identify such markers in human populations. And as you know, both in Europe and the United States, there are some large cohorts of individuals whose medical records have been available for the last 25 to 30 years. And these individuals have donated blood every 2 years during the time in which they have been enrolled in those types of things. So that means that if you identify a patient which is a member of this cohort who has developed a cancer, that they have contributed their blood and therefore plasma samples at least 2 years prior to that. So we were able to obtain such samples, from individuals. So essentially, you're sampling individuals, plasma samples before they were diagnosed with cancer, and use mass spectrometric analysis to be able to do that. And here is one such study that was done with lung cancer. And in this particular case, we've used many different types of genetically engineered mouse models and patients, and we were able to identify a number of different markers that actually were found to be elevated in individuals or in these models, prior to developing cancer. And so these types of studies also revealed, in the case of colon cancer, a variety of different markers. And I'll just give you one example of one marker that we identified, and that marker is called cathepsin D, a protease that is found to be increased in colon cancer patients before they were diagnosed with colon cancer, as shown here. And so we're actually able to develop a reagent to try to detect the presence of cathepsin D, and to be able to do that, this actually illustrates some of the complexities that are involved in actually clinical development of these types of things. But first, you developed a reagent, which is a fluorescent reagent, which is normally quenched, and this fluorescent reagent can be injected into humans or mice, and they would go throughout the body. But that reagent would be cleaved only in those cells in which the cathepsin D is highly expressed. And when it is cleaved, the quenching is relieved, and you'll be able to see the fluorescence. And here's an example of that in a mouse model that has tumors. And as you can see, the left-hand side of the image is just white light, and the right-hand side is the image that was obtained at 690 nanometer wavelength. So this particular reagent is now entering into clinical trials at MGH to test them in patients. But this sort of caused unique types of problems, even though we're able to identify this particular marker, and it can be used for this purpose. First of all, in the case of human colonoscopy, all of the colonoscopies are done with white light and no other wavelengths, and we had to develop a new type of colonoscope that is actually capable of looking at different wavelengths. So that's actually very difficult to do, and we had to convince some of the imaging people to develop these types of things. And the second is that if you want to be able to develop the reagents, then you had to work with the diagnostics companies to be able to validate these types of markers and produce them in GMP facilities, to be made available for patients throughout. But nevertheless, this sort of illustrates how it is possible to actually take these types of protein-based biomarkers. And there are large numbers of such protein-based biomarkers that are now been validated in many different cancers, in colon cancer and lung cancer, ovarian cancer, and many other types of cancers. And it is indeed possible for us to utilize those reagents. Another approach that we have also heard about today is liquid biopsy. And many of these tumors, they have either cells that go into circulation or the DNA, the degradation of the cells that get into the circulation. And it turns out that approximately 1% or less of the DNA in the circulating plasma could be from tumor samples. And so the technologies have been developed to be able to just look at these so-called liquid biopsies and use them to ascertain whether the tumor sequences are present. We've heard some of the approaches this morning about the complexities that are associated with this approach. And the complexities are first that the amount of tumor DNA that is present in circulation is not necessarily very high in all cases. And the second is that, because of the tumor heterogeneity, the depth at which you need to be able to do the sequencing of this DNA from circulation is significantly higher, therefore more costly. But nevertheless, this approach is now being used certainly to be able to monitor patients and their response to therapy, but it's also potentially can be used to be able to identify people who have had a tumor but has not been clinically diagnosed to have a tumor. So there is a new research efforts that are currently underway to see whether it is possible to use liquid biopsies as a screening tool for them. And finally, what are the kinds of approaches that we're using for treatment? And then we heard many different examples of those. And Stefan talked about how things have changed in the case of lung cancer over the last 10 years or so. And clearly, since 2004, when the EGFR tyrosine kinase inhibitors were approved for therapy, in these last 10 years, that our ability to be able to look at sequencing, these tumors, and we have heard different types of panels of sequencing, 400 genes or 600 genes, and actually it turns out that many people think that, we might miss many other mutations if we use these panels, but in fact, there have been studies that have been done that using these panels of 400 or 600 cancer genes actually is equivalent to sequencing the entire exome of the genome. So the results are very reliable, and you'd be able to classify these patients into all of these molecular categories. And certainly in the case of lung cancer, as we know, that there is either a drug that's available for all of these different types of rearrangements that are approved for non-small cell lung cancer or currently in clinical development. Other types of things that are happening are really very exciting and interesting, but also pose unique challenges. This is an example of a study that we have done a few years ago on colon cancer. And when we examined a large number of colorectal tumors, we discovered that about 4% of those tumors have amplification in ERBB2 or HER2. And as all of you know, that the Herceptin was approved in 1998 for breast cancer patients who have overexpression of HER2, and that drug is quite safe and effective in that subgroup of patients that contain such amplification. So the idea is that can we test these patients in colon cancer that have these types of changes? But what's really interesting is that these types of, in the case of HER2, the amplification is not restricted to breast or colon cancer. And here there are 40 different types of cancers. A proportion of tumors in all of those cancers have amplification of ERBB2. Now, this poses a very unique opportunity and challenge. The opportunity is that we already have a drug that is known to be safe and it is effective for this type of a change, and therefore one would imagine that those drugs could effectively be used here. The challenge is how do we do a clinical trial? All of the clinical trials that are currently done, for these, even for such a drug as this one, is that you need to be able to do that at one cancer at a time. And that paradigm, as you have heard this morning, is not sustainable, and we need to think about other ways in which we'd be able to use this. Is it possible to conceive of a clinical trial that is based upon the genetic constitution of the tumor? There are issues with that because it turns out that even though the same mutation is present in different cancers, the different types of cancers from different tissues of origin don't necessarily react in exactly the same fashion. But do we just throw the baby out with the bathwater, or do we think about a way in which we'd be able to solve this problem, which we need to be able to do? The same thing is true with other types of things. We have heard this morning about PARP inhibitors that have been approved for ovarian cancer patients who have germline mutations in BRCA1. But it doesn't matter whether the tumor has a mutation germline or somatic, because the PARP inhibitors work in those tumors or in those cells in which the double-strand break repair pathway is impaired. And now it turns out the same types of genetic changes are present in a variety of other tumors. Here is an example of pancreatic cancer. On the right-hand side, all of those different genes, the RAD54 or the ATM, and BRCA1 and BRCA2. Now, if you look at all of them collectively, there are a large number of genes that are mutated, right? A large proportion of the tumors are mutated in one of these genes that impairs double-strand break repair. So that means that all of these patients can potentially benefit from PARP inhibitors, but we need to show that to be the case. Similarly, in the case of DNA repair, again, in the case of colon cancer, it has been shown that those patients that have DNA repair deficiency are the groups of patients that respond very effectively for the immune checkpoint therapies, whereas individuals whose tumors do not have such changes do not respond at all. So such changes are present in many, many different tumor types, and we need to think about how we would be able to bring those approaches to all of these different tumors. And finally, we talked a little bit, Stefan talked about mutation burden, and this is what I talked about in the case of colon cancer. There are a group of tumors, a large number of them, nearly 85% of the tumors or so, that have on the average 1 to 10 mutations per megabase of DNA that is sequenced, and there are a subset of them, the remaining 10-15% of them, which have mutations which range from 10 to 1,000 mutations per megabase of DNA that is sequenced, and it turns out that all of those tumors that have this high mutation burden are the ones and the only ones that respond to immune checkpoint therapy. And about these immune checkpoint therapies that are emerging, and here are actual results from the colon cancer results from the clinical testing, that all of those lines in the blue are the ones that have mismatch repair deficiencies, and those are the ones that respond very, very effectively to the cancer. So given all of this type of information, what is the sort of outlook that we think about? I would conclude by saying that the scientific knowledge about the role of genetics in many diseases, certainly including cancer, is changing dramatically. And this result directly is leading to the development of new drugs and therapies. And certainly from my perspective as a geneticist, the reductions in the cost of DNA sequencing is really making all of this genetic testing affordable. And I would imagine this will continue to be the case. But to be able to facilitate all of this information, we need to be thinking about new approaches for clinical trials. And thank you very much.
This morning, we had in 2 times liquid biopsy as a key point. So I do have a question. One source of information is circulating DNA, but an alternative source of information can be CTC, cancer tumor cells. And in your view, there are differences in the two, and the information we can get from circulating DNA, in my view, is information of the median mutation situation. Why maybe circulating cells may give us information about metastatic potential. And so it can be a simplification of the heterogeneity of the results we can get from liquid biopsy.
Yeah.
I want to have your opinion.
So first of all, with regard to circulating tumor cells, there have been a lot of studies that have been done, and so that it is indeed possible to be able to have circulating tumor cells. But the circulating tumor cell studies have really came before the knowledge that we have circulating tumor DNA, and and in a way, being able to look at the DNA directly, that circumvents the need to isolate circulating tumor cells and analyzing them. And actually, Stefan showed some results this morning with circulating tumor DNA with large panels of genes that you could test for them, and how well it represents, the actual tumor. I think the only issue is that if the amount of DNA, whether it is coming from circulating tumor cells or just direct DNA, is what proportion of the total DNA is the tumor DNA. And that usually is an issue because the amount of tumor DNA that is present in circulating plasma is only about 1% or sometimes less than that. And that means that you need to be able to do, if you're going to use sequencing, to do significant in-depth sequencing to be able to identify the mutations. But I think that there are more studies that are coming out from now, and that would validate this approach further. You might want to make a comment.
Yeah, maybe just to quickly add, I mean, the difference between CTCs and ctDNA is in a way, I mean, what we look at, let's start from a different point. If you look at CTCs, there are more than 16,000 papers published on CTCs. None of them have been predictive. How many more do we need? So the question here is in our point kind of, in a way, not simple, but relatively straightforward. We believe, as Raju said, I mean, the devil is in the detail, and I will talk about it in a minute. But the question is ctDNA more comprehensive, reflecting what's going on in the tumor than CTCs are? And that seems to be the case. I'm not denying that CTCs have a value in being prognostic, and they certainly have shown it. I mean, no doubt about it. The question is, can it be predictive, and can it be comprehensive? And that's the point. But CTCs is something I think we can still learn a lot from it by applying different technologies, as Raju also said. And for the ctDNA, the question is sensitivity. Absolutely. The question is, how deep is deep enough? What allelic frequency do you need? 1%, 0.1%, 0.5%? And the question is, what allelic frequency sensitivity do you want at a baseline diagnostic in order to do screening? As Raju said, I mean, there's not, in adjuvant tumors, there's little to nothing sometimes of ctDNA you can measure. And the question is, can the technologies become better, or maybe it is really not accessible in an adjuvant cancer because not so much DNA is shed. So we look at it on a more, is it comprehensively better than a CTC? That's, we believe, yes. But is it something for all stages of the tumor? Maybe not. Yeah. Thank you. Yeah, there's another one.
One of the strategies for PGG is that for the drug development. I mean, we know that this is highly costly, and how can you reduce, what is the strategy for PGG to reduce the drug development? We know that it takes 10 years if you get lucky for oncological medicine, 10 to 12 years. And the second question, if I may, to maybe all the speakers,
Yeah.
The drug development speed and the cost effectiveness. I mean, all these technology and NGS, we know that it's really costly, and it's really expensive. You know, like it takes probably costs about 100,000 euros for a cancer patient to get treated. How can we reduce this with these new technologies and all the NGS plans that you highlighted? Thank you.
Thank you. You want to start?
This doctor.
Okay. So, I mean, the question basically in terms of how can we change the speed and how can we basically do drug development faster, better, more efficient, I think it's definitely something we need to look into that's part of this IMI work, how all the companies can come together and can do it. We believe one tool, and I would really ask also David and Alexandra to comment from a clinical perspective. So what we believe is we change the way we do clinical trials from we do large, global, thousands of patient trials, and then we try to learn something of it. If we change into smaller but data-rich trials, what I mean with data-rich is really we have sequences of biopsies, we have sequences of data capturing, we do multiple imagings, and so on and so forth, and not kind of only at baseline and then of end of treatment. So there's a huge big hole in between of the data we see in clinical trials and we don't understand. So I think we need to come to, let's say, a 20-patient trial, a 50-patient trial. We may learn more out of such trials than out of 200 patients. And the question is then basically how we can expand it and what is evidence enough to get a registration. And that's what we need to talk with regulators, so to say. What do they really constitute as clinical benefit? That's why I talked about changing clinical endpoints to something which is more tangible, but also, of course, it needs to be safe and it needs to be proven that it works. But we believe if we do it in a kind of a more way of rigorous, smaller trials, and Raju basically said, HER2 is something which is not only present in breast cancer. You see it in colon, you see it in bladder, you see it in many other trials. Why is it not registered? Because nobody runs 10 clinical trials, so to say, in thousands or hundreds of patients. But maybe David and, yeah.
Well, I think we're facing a number of problems all at the same time that we're not necessarily going to resolve all at the same time. But on the one hand, we see fragmentation by lead mutations. But the premise that a certain mutation would have the same importance across different tumor types has already been elucidated. It's not the case. But a very, very pregnant example is BRAF mutation in melanoma. If you have a BRAF inhibitor, you get an important, but in general short-lived, very important response. Let's say on average durability 5-6 months. But it's true for 50-60% of those melanoma patients. Exactly the same mutation in colorectal cancer exists. The response rate is 5%. So it's the same mutation, but it has a completely different meaning in the context situation of different organs. So organs are like countries, and they have their own culture, and a cup of coffee in the UK is not like a cup of coffee in Napoli, because Napoli has the best coffee in Italy. So, you know, it's like heaven. So this is the real world. So the real world also means that you know, thinking that we can resolve everything by taking true-cut biopsies out of tumors is not necessarily true either. So you stick a needle into Europe. Now, your needle lands either in Zurich or in Macedonia. So what's your conclusion about the tumor? So that is how heterogeneous, and actually in the USA, when we look at the election primaries, I would say you have had some heterogeneity in the USA as well. So, meaning that dealing with heterogeneity is phenomenally difficult. Then the question about what overall readout system circulating DNA gives out of the blood is therefore a very important one, but it's got to be at some point not only a selection of tumor load, but also of various aspects of tumor biology. So if it's a key aspect of tumor biology that's represented in the circulating DNA, you may even pick it up at an early adjuvant setting and it may be of importance. But if it's not, because that's not what's circulating, for whatever biologic reasons that are behind it, because it's not shedded, it's not, etc., then you pick up false signals. You say, I see all sorts of stuff, but you will not get beyond prognostic. For sure, you will never get beyond prognostic. So we said it's the end of blockbusters because of all this fragmentation, molecular alterations, and all these things. Then we try to say, yeah, but it's not so bad because if you have a molecular alterations, it will be true across tumor types. Sorry, it's not true. Woo. This is a little bit of there's a bit of a party pooper because we thought that molecular medicine was going to solve it, but then we found out that organ is even more important than et cetera, et cetera. And then in immunotherapy, because now you're dealing with a functional system that has a completely different readout of what are tumors, how we recognize it, how we fight it, we now have the biggest blockbusters in the history of cancer medicine, and it's called anti-PD-1. So there's nothing, so this is now the most important drug in the history of cancer medicine. So actually, we are not going to see any more blockbusters. No, why not? There can be yet another anti-PD-1. Because only 2 key molecules have changed the whole face of the earth in systemic therapies, and it was the discovery of CTLA-4 and PD-1. So who tells me that there's not out there a third molecule that will be also of that kind of an importance? So in the end, the big shifts are by biological new ideas, and the incremental shifts are what high tech allows us to put on top of it in terms of incremental development. And regarding pricing, I'm always surprised that the discussion is as if we accept that you have to pay 100,000 euros per year or 160,000 euros per year, or for the Epi-Nivo combo, $256,000 per year. I mean, what kind of a point of departure for a discussion is that? So I think we need to change the point of departure. And if we're not going to have reasonable regulation on drug pricing, success will kill the whole field. So I don't see the interest of killing the whole field by not talking about that the fact that the point of departure needs to be changed, and not how can we pay all this money. No, I would say stop paying and see what happens.
I shall not disagree with Lex, because I think it's a very valid point. But why do companies expect and sometimes have to charge that price? Because of a very wasteful drug development process. How many companies are developing PD-1, PD-L1 drugs in solid tumors? 4, 5 companies? They will all be running a randomized trial with a control arm. Most of those control arm patients are wasted, because we repeat the same question with different drugs. Can we not develop a system where you can use the same control patients for each of the different companies' drugs? Then you need fewer patients, you get the answer earlier. It takes a mindset to think differently. Each little silo will do its own regulatory study with its own set of control patients in its own collection of institutions. That's wasteful. And ultimately, to me, it's wasteful of patients, because we're putting more patients on a control therapy than we actually need to answer the question. And the same thinking can be drilled back into the earlier phase 1, phase 2 go, no-go studies. Take those 4 or 5 companies developing anti-PD-1, PD-L1 drugs. what consistency do we have about the level of PD-1 in the tumor as the entry criteria? So there you are. We've created a system which is designed to drive up costs. So let's change the system to bring the costs down.
Denny,
What I want to say is that all what we hear, the diagnoses have been made. I think we are here, at least at IMI, to find solution to that. I try to depict in my presentation that through this type of new platforms, for instance, what David pointed out just now, which is shared control population, can be addressed. So I think the diagnoses have been made. We have some solutions that, for instance, EURTC has been piloting. Cancer Core Europe has been piloting complementary solutions. How are we going to use these solutions? And I think there are people in Europe that can bring this solution. How do we get together? It's not a matter of technologies. It's not a matter, or I would say, of organization. It's complex, but we can do it. I think it's a matter of will. That's it. And if I came to this meeting, it's because I think there is matter, there are solutions existing. How are we going to put these solutions together in motion? Because we can say until tomorrow there is a need for a shared control population. But okay, we know it. How do we make it happen? That's the next question.
Yeah, I mean, just quickly to that, I mean, this is what I said in the morning. When we talk about real data, I mean, I do agree with you. I mean, if garbage in, garbage out, so to say, when the big data, so to say, we put in are really not well annotated, curated, and there's nothing coming out of it, that's clear. But can we not do it also and have a kind of a virtual control arm? Do we even need one? If we do know what a normal PD-1 in lung cancer is performing, and why do we even need it? And those things is something we need to think about it. You know, if we have the data in a very concerted action, so to say, from all over the places and in one disease or in many diseases, why we would even do it again and again and again, you know, and that is a cost driver. And at the end of the day, to your cost question, I'm not a pricing person or something, but I think things are changing. I mean, we come more to the part that we pay for value and what really does it. I firmly believe the thing needs to change. If we really go to that, we change the treatments more dynamically. What you're going to do? Pay for 3 months of PD-1 and pay then for 5 months of BRAF and pay then for 3 months of whatever. There needs to be different solutions because that's absolutely not feasible. Yeah. There was a question here in the front row.
Yeah, I was just making the point that
Okay.
the combination therapies and how that can be dealt with has been already invented in the late 90s for a different field called HIV.
Absolutely.
And I think it would be interesting to review how that process has worked to bring industry together to find the best treatment for the patients. And this model could be there. And the other question or point which was that there's a lot of big gaps in data collected, and this has also been seen in other fields, the biobanking, where it has been realized that the old biobanks have no value because you cannot put them together because you have no common point, so you need to restart from scratch with all the new information. And I think now with the strength of the SAP system, you will even find more gaps in the old system. So 2 lessons which can be learned from different fields.
Nice.
That's actually a great point. As you know, certainly in the United States, the reason for all of those types of tests was based on advocacy. And the patient advocacy really a great difference in sort of really developing all those combination therapies for HIVs. So we should have something comparable to that. But I also wanted to raise one issue that we haven't talked about just a little bit today, and that is the following. Is that the part of the difficulty, the time that it takes to be able to develop the drugs, and the cost of developing the drugs is cost of clinical trials. And all of the clinical trials, and all the ones that we talked about this morning, and certainly in the United States, most of the clinical trials are done in academic medical centers. But certainly in the United States, 80% of the patients are not treated in academic medical centers, but are treated in community. How do we access all of those patients that truly would be able to benefit by entering into clinical trials, but they do not do so? And the issue is they don't want to go, you know, from someplace in rural Germany to Heidelberg, right? They don't want to go. They want to stay where they are. So one of the ideas is, instead of sort of having clinical trials that patients go to, can we send clinical trials to the patients? I mean, we talked to Stefan about that, but perhaps, some of you who are actually involved in enhancing the opportunities to provide clinical trials, maybe you can comment about what are the kinds of things that we could do to increase the number, and that would significantly reduce the time for enrollment and approval of drugs.
David?
So I can make a comment from the UK because we have government-funded clinical trial infrastructure across the whole of the UK. And if we look at our recruitment to cancer studies, they're not all randomized trials, that rose 4 or 5 times, not 4 or 5%, 4 or 5-fold over a 15-year period. So that, for example, at the moment in the UK, we recruit between 40 and 50,000 cancer patients a year into clinical studies. However, we do not recruit 40 or 50,000 into registration studies. Why? Because the registration studies are managed by pharma multinationally, and on the whole, with some exceptions, those very large commercial registration studies that need total dossiers to go to a regulator will want to run in a small number of centers in each country. Whereas we can run an academic study, and I chaired a UK breast cancer study, which we opened in 120 hospitals in one country. So again, it's about a mindset change. If you want to get access to the whole country's population of cancer patients, we need to rethink the model of a regulatory study. Why do you want to only run it in 6 or 10 centers per country? Because of insurance costs, because of SDV costs, et cetera, et cetera, et cetera. But actually, how much of that is necessary in the same way? Because I agree, patients will sometimes travel to Heidelberg, but actually there is a political imperative that patients should be treated equally well with equal access to clinical research where they live. So how do we change this model of forcing the patient to move to the study? How do we change so that the study can reach out to the patients? And I am involved in some conversations with the MHRA in the UK at the moment around what we can do within the regulatory process, but that's largely for academic studies. It gets more complex for a regulatory study in the current framework. Denis, do you want to comment? Oh, sorry.
I think Denis and I, but you will have additional points certainly to what I'm going to say. There are, however, different worlds of clinical research. And the early clinical phase studies, I'm sorry, it's not going to happen in the peripheral hospitals in the country. There is no culture there of doing clinical research at all, let alone doing and addressing the biomedical experimentation that's ongoing in early clinical trials. So that's where the real innovation is, because let's face it, once you have reached the randomized phase 3 trial structure, it is widely applicable. And so right now, you have phase 3 trials with 350 centers. That means actually that you're going into also the larger peripheral hospitals for those kind of trials, even for pivotal trials. So you have different layers of what needs to be done. And the attrition rate that is happening in the early phase trials, if you let a drug through to a phase 3 on flimsy arguments and on a lot of hope and rolling the dice, yeah, of course, then you're going to launch it into a $300 million losing proposition, et cetera. But the major attrition rate, of course, is in early clinical trials where things are not passing the bar that should be raised all the time, so that your proof of concept studies actually know that you now are in control, in understanding, and you can take it all the way. And so we know that over the last 20 years, before the last 5 years, we've been much too lenient by letting drugs onto the market, which benefit was marginal at best, if any at all. And that was all wasted money to pay for those drugs. But it set actually the stage to now triple the price for some things that work, and that may be actually hyped also a little bit. But this hyping leads now to a totally unacceptable and non-sustainable situation. So if we do not have a much more stringent passing the bar and attrition rate system up front before the big trials are launched, well, yeah, then we can simply continue to waste money. But there is an interest to keep all this machinery going. So there's also people who actually are quite happy to keep it going like this. It's like having a private prisoner system, you know, where you end up with 1.3 million people in prison because it's a private enterprise. There are parallels. We live always in parallel worlds. I'm not accusing you of anything, but it's just an observation that we all have different problems.
In one of my slides, you have seen that transformation needs two routes. I said there is one route that I'm not going to develop, which is the operational and regulatory route, okay? And I decided during my presentation to focus on the methodological benefit of the platform. Actually, your question related to community center is in the other route, and we have been questioning that, and we have been looking into solutions. And I think what Lex just said is important, because first we have to a little bit clean the field, indeed, of what type of clinical trial we are doing, but that's the first thing. And you've seen also my slide with all this embolization in oncology, so I think there is a need to a little bit address the chaos. That's the first thing. The second thing, we see also different type of clinical trials coming. And for instance, there are some clinical trials, even in early development, that are potentially amenable to community sites. And I think we have to see a little bit what we do in the big centers and what we can bring into the community center. But the next challenge, and that's the challenge that industry has brought to us, is if you want to bring the clinical trial to a patient because he's in the community center and he has this rare alteration that you are looking for, then you need to bring the drug supplies there, the new drug. And that's a major issue. And I think in the process that I would like to start maybe as an outcome of this forum is actually how also we address these things. So we are discussing that actively, because it must have an impact on the regulatory framework and the operational framework, but it's true, and that's why I put the two routes into the slide. If you want to win the battle, you have to know your battlefield. And if you don't take the two arms together, then you are not going to make it. And that's where I think, for instance, infrastructures that have the expertise, we can get together and we can try to address that.